Okay, so this is our second part of what we talked about in class today. We're going to talk about credibility. So this is what we look at to know whether or not an experiment was well designed, or whether or not a theory holds water. We're going to be looking for these aspects in a particular experiment or set of experiments in order to understand whether or not they're trustworthy. So the first thing we're going to look for is that the experiment is repeated. So I should be able to get the same results that you get. I should be able to get the same results over and over again. If I can't get those same results over and over again, or if nobody else can get those same results, then that indicates that there's probably something wrong with the experiment. The next thing that we look at is sample size, and this is actually one way we look at whether or not we have repeated the experiment. So for chemistry or physics, we might just have a handful of trials, maybe 3 trials or 5 trials to see, is this something that is repeatable? But for things that involve living beings, there are so many outside influencing factors that can impact our results, be they the temperature, the environment, the culture of an area, the male versus female versus non-binary, the gender of a particular person or group, the age of a person, the socioeconomic status. All of those things can influence or have the potential to influence the results. And so we want to have a large sample size that includes as much diversity in all of these areas as possible. But we want to keep in mind that whatever sample we look at, that is going to limit who our results apply to. So if I only do my study on college-age males, then my results only apply to college-age males. And it might be interesting to see if they also apply to other people by doing a further study. So I might say, oh, this merits further study, but it doesn't necessarily mean my results apply to that. So how much is enough? How much is a lot? Generally, if I'm doing a study, say on a drug, I'm going to start out with just a handful of people in order to see, is this worth investigating further? Because if there are problems with it, I don't want to cause problems for thousands and thousands of people. And if everything looks okay with that very small group, then I might expand my next experiment to include hundreds of people. And so with this, I'm looking for efficacy. Does it work? And I'm also looking for safety. So I'm going to be asking these people, what are the potential side effects that you have with this? Are you experiencing headaches, sleeplessness? Are you experiencing soreness? All of those things that you hear in the commercials, that this may cause such and such and such. This is where they start looking for those potential side effects. But in order for the results of the study to be truly credible, we want to have thousands of people in the study. So if you're looking at a scientific study and there's hundreds of people, we might say, okay, that's really interesting. This merits further study. But we're not going to say, oh, definitely, this is definitely true. Because with hundreds of people, you're not necessarily going to get the whole diversity of all people everywhere. We're not going to have the randomization of people from all different continents or all different parts of the United States, all ages, all social backgrounds, all genders. We're not going to have the diversity to say, yes, this is something that applies to everybody. So we really want to have thousands. And even as such, we're probably only going to have that within the geographic region that we're studying. If I'm a scientist in the United States, I'm probably going to have subjects that are Americans. If I'm a scientist in France, I'm going to have subjects that are French. If I'm a scientist in South Africa, my subjects are probably going to be South African. So we need to remember that whatever conclusions we draw, whatever results come from this, they're only applying to that particular group. So if somebody does a study in South Africa and it appears that for people who live in South Africa, there is this connection between A and B, then as a scientist in France, I might say, I wonder if that's also true for people in France or in Europe. I'd do a study there. And a scientist in China might say, hey, it looks like it's true in South Africa and in France. Let me try it here. And in the Americas, you might have people in different countries trying it. And the more our results are repeated everywhere and applied to different populations, the more that we have credibility. So another thing that we can do to increase our certainty in a particular experiment is to have a control. This is sometimes called a control variable. I don't like that because the control is the thing you're keeping the same or constant rather than something that changes. So the terminology doesn't quite work that well. But what we might do is we might say, what happens if we do nothing? So if I'm testing a drug, I might have two groups, and one group gets the vaccine for COVID and the other group gets a placebo. So the placebo is sometimes called a sugar pill, but it wouldn't be a pill in the case of a vaccine. It would be a shot that looks like it's a vaccine, but doesn't have any of the vaccine material in there. Maybe it's salt water, something that you wouldn't know the difference. And so the person who is receiving the shot, and often the scientists themselves, don't know whether or not that person has received the actual vaccine. I can also do a scenario where if I'm trying to test whether one thing influences another, I can say, okay, what if I don't apply that independent variable? So I can have, maybe I'm testing a fertilizer on different trees. I can have a little bit of fertilizer, a medium amount of fertilizer, a lot of fertilizer, but I also want to see what happens if I don't have any fertilizer at all. So that would be my control scenario. This is what it looks like if we don't do anything. So let's talk a little bit about some variables. The variables are the differences between the groups. And generally what we're looking for is some sort of relationship between the variables that we are studying. So we might have a relationship that we call a correlation. So a correlation often means that they trend together. As this increases, this also increases. So for example, the older or the taller somebody is, the bigger their feet tend to be. So there's a correlation between your shoe size and your height. But that doesn't mean that one causes the other. Having big feet doesn't necessarily make you tall, and being tall doesn't necessarily give you big feet. So for example, I have a sister-in-law who's almost the same size as me, who has very teeny feet. She's about 5 sizes smaller than I am. And even though we're about the same height. In contrast, my own sister is about 4 inches shorter than me. And so she's significantly smaller, but she has the same size shoes. So size and shoe size don't necessarily one cause the other. So having big feet doesn't cause you to be tall. Being tall doesn't necessarily cause you to have big feet. What is going on is that there is a relationship where they are both related to the same thing. So I'm going to give you a little bit more of an obvious example. In the summertime, the temperature tends to go very, very high. And when that happens, ice cream sales increase a lot more ice cream sold during the summer than in the winter. Another thing that happens during the summer is that there is a higher crime rate. So we wouldn't try to say that crime causes people to eat ice cream, and we wouldn't say ice cream causes crime. That doesn't make a lot of sense. They are correlated to one another because they're both related to this variable of the outdoor temperature. So sometimes we know what that correlating variable is, and sometimes we don't. We can just say, hey, they seem to be working together. So sometimes in studies, you'll see these are correlated. We don't know if there is a causal relationship. So let's talk about that, causality. Causality means one thing causes the other. So an example of this might be if we look at height, and instead of shoe size, we look at age. So if I look at 1-year-olds and 3-year-olds and 5-year-olds and 10-year-olds, I'm going to find that the 10-year-olds by and large are larger or taller than the 5-year-olds, and they're going to be larger than the 3-year-olds, and they're going to be larger than the 1-year-olds. And so in that case, there's definitely that correlation, but we can also say there's a causal relationship. For people 10 and under, the older you are, the taller you are, because time allows you the chance to grow. So that age is a causal relationship with the height of the person. Okay, so we've described in there things that are different between different people. We've talked about their size, their shoe size, their age. Some of these are independent variables and some of them are dependent variables. So when I'm conducting a scientific study, an independent variable is the thing that I think is causing something else to happen. So this thing is usually what the scientists, before the experiment ever starts, makes different between the groups. So they're going to say, okay, I want to look at how age impacts these things. So I'm going to divide my subjects up into different ages. Or maybe in the case of our coronavirus, I want to see whether the vaccine or no vaccine or 2 shots of the vaccine makes a difference in whether or not people get the coronavirus. Okay, so that's something that is set up ahead of time. The scientist says, these are going to be my groups. Okay. This independent variable is going to theoretically, that's why we're doing the test, we don't know if it's going to happen. Theoretically, it is going to cause the dependent variable. Now, with the dependent variable, I may have more than one thing that I'm observing. So these are the differences that are measured by the scientist. So in the case of our age versus our height, the height is the dependent variable. Our height depends on our age. In the case of the vaccine trial, the whether or not somebody gets the coronavirus is the dependent variable. So that's the main thing that we are looking at in that case. But I can look at more than one dependent variable. As long as I only have one independent variable, I can have multiple dependent variables. So maybe I'm also looking at how severe somebody's symptoms are if they do get the coronavirus. So maybe I find that there's not a difference in how much people get it, but their symptoms are a lot less. And maybe I'm also looking at the side effects. Maybe I'm also looking at the ability for people to spread that disease. And so when you read about the coronavirus, you're going to read how they tested these different areas and found different levels of causality between having the vaccine and each of these dependent variables. Okay, so key thing here is the dependent variable is what we're measuring during the experiment. What we're seeing, did this result from our independent variable? All right, so generally when we have a hypothesis, our hypothesis is written so that we're saying the independent variable causes the dependent variable. Okay, so that's why it's the dependent variable, because it depends on the independent variable. We can only have one independent variable at a time. So if you're looking at a scientific study and it looks like one group has 2 different independent variables, maybe I have one group that they're getting placebo or not the real vaccine, and I have another group that's getting the vaccine, but I also have the people who are not getting the vaccine are told don't wear a mask, and the people who are getting a vaccine are told do wear a mask. There are 2 differences between those groups before I even start measuring my dependent variables of whether or not they received or whether or not they contracted the virus. So we need to check to make sure that there's only one independent variable at a time. Now, we'll talk later about how there are times where we might have what I might call a complex study, where you have more than 2 groups. And if you have more than 2 groups, as long as the differences between the groups that you're comparing are only one thing, then you're fine. So for example, maybe I have a group that is placebo, no masks, and I have a group that's placebo with masks. And then I have a group that is vaccine, no mask, and a group that is vaccine with masks. And so I'm doing all of those experiments at the same time, but I'm only comparing the placebo group with no masks to the vaccine group with no masks, and I'm only comparing the placebo group with masks to the vaccine group with masks. All right, so that kind of wraps up what we talked about today. Hopefully that keeps you caught up on what we have been doing, and I hope to see you in class soon.